Welcome back to the deep dive. Today, we are taking a monumental leap into what you might call the ultimate biological building material, the human skeleton. We're aiming to move far past those simple anatomy diagrams you might remember. We're plunging straight into the cellular chemistry, the dynamic mechanics, and frankly, the evolutionary genius that defines osseous tissue. The sources we've compiled for this, and these are straight from the foundational textbooks that are really designed to give you, our listener, an elite professional level of mastery. We want you to master the structure, the function, the dynamic development. We're focusing on every single concept provided in our source material, making sure nothing's left behind.
Exactly. And when we think about bone, it's essential to immediately recognize its duality. You've got two major roles happening simultaneously. First, the obvious ones, the mechanical functions, providing that solid framework, resisting gravity, protecting incredibly vital structures like your brain, your lungs, and of course serving as that system of rigid levers that your skeletal muscles need to actually create movement, locomotion.
Right, the scaffolding and the protection.
But the truly fascinating element, and maybe the more complex one, is the metabolic function. Bone is just this massive storehouse for vital elements. It's the master reservoir for calcium, phosphate, other critical ions. It's an active metabolic organ, constantly monitoring, constantly adjusting these stores to maintain those precise, narrow chemical concentrations needed everywhere else in the body.
So it's not just inert scaffolding.
Not at all. And mastering this subject really means understanding that this seemingly static architecture is governed by a fiercely dynamic, highly responsive cellular population. It's a living structure, constantly being, torn down and rebuilt.
Okay, let's unpack this then, starting right at the fundamental chemical ingredients, the matrix itself.
Sounds good.
So what gives bone its signature hardness? It all comes down to the matrix, right?
Yeah.
And it's this brilliant partnership between the hard mineral part and the tough organic frame.
That's the essence of it. Chemically, this partnership is roughly half and half if you look at the dry weight. So 50% is the inorganic component. And this is dominated by crystalline calcium phosphate, specifically in the form of hydroxyapatite. It's a highly structured mineral. Chemically, you often see it written as text EAF2.
But it's not just pure hydroxyapatite, is it? There are other ions mixed in.
No, exactly. It's not pure. Alongside the calcium and phosphate, you find bicarbonate, citrate, magnesium, potassium, sodium ions. They're all embedded within that crystal lattice. And what's absolutely critical here, something often overlooked, is that the surface of these hydroxyapatite crystals is highly hydrated. There's a layer of water molecules bound to it.
Okay, why is that hydrated surface layer so important? How does that impact its function?
That's the key to its role as an immediate buffer system. That thin layer of water facilitates really rapid ion exchange between the mineral store and the surrounding body fluids. So if your blood calcium levels start to dip, even slightly, this hydrated surface acts as the quick response system. It can immediately release some calcium ions to help stabilize things before the slower, cell-driven mechanism involving osteoclasts even need to kick in.
Fascinating, a rapid buffer. Okay, so that handles the hardness, the mineralified. What about the other 50%, the organic component, the osteoid?
Right, the osteoid. This makes up the remaining 50% of the matrix by dry weight, and the vast majority of this, about 90%, is type I collagen. This is the same collagen type known for its exceptional tensile strength in tendons and skin. It forms the flexible framework.
And it's not just collagen alone.
No, it's supported by a collection of smaller molecules, small proteoglycans, and key multi-adhesive glycoproteins. A key one is osteonectin, which acts like a bridge, helping to link the organic collagen fibers to the inorganic mineral crystals.
So this composite nature is really the secret sauce. If we were to experimentally remove one component, the function of the other becomes clear.
Exactly. It's laid bare. The mineral part provides the hardness, the resistance to compression that lets you stand up. But the collagen network provides the pliability, the tensile strength, the ability to absorb shock without just shattering.
Like if you decalcify a bone.
Right. If you treat bone with acid to remove the mineral, decalcify it, the overall shape remains, but the structure becomes soft, flexible, almost rubbery. It proves that the resilient collagen network is holding that architectural blueprint together.
And the opposite, if you remove the collagen.
If you were to, say, burn off the organic component, you're left with the mineral structure, which is hard, yes, but incredibly brittle. It would shatter easily. You absolutely need both components working together.
And here's a really profound consequence of that calcification, that hardness. Nutrients in waste products can't just diffuse through this solid matrix like they do in other tissues.
Precisely. And that limitation dictates everything about how the cells within the bone survive. They have to evolve this unique, incredibly complex communication network just to stay alive and function. Which brings us neatly to the three key cellular players.
Okay, let's start with the builders. The osteoblasts, these are the architects, right? Where do they come from?
They originate from mesenchymal stem cells, those multipotent progenitor cells found in connective tissue. And their primary function, as you said, is secretion and construction. They build the bone matrix.
Their whole mission is to synthesize and secrete the organic parts, the collagen, the glycoproteins, that initial unmineralized matrix we call osteoid.
Exactly. And you find them exclusively on the surfaces of existing bone matrix, or where bone is about to form. They typically arrange themselves into a single, continuous layer of cuboidal-shaped cells.
And they're connected.
Oh yes, very tightly. They're linked by gap junctions and adherent junctions. This allows them to communicate and coordinate their activity as a functional sheet ready to build.
Now, let's get into the real elite-level detail here. The precise molecular steps they take to turn that soft osteoid into rock-hard bone. Mineralization isn't just passive, is it?
Not at all. It's a highly regulated, active process involving some very specialized molecular tools deployed by the osteoblasts.
What are the key players?
Okay, two crucial components. First, they produce a protein called osteocalcin. This is a polypeptide whose synthesis depends on vitamin K. Osteocalcin has a powerful affinity for calcium ions, Text-T2 plus 10-DAR, binding them locally and effectively concentrating them.
Creating a calcium-rich zone.
Exactly. And even more critically, they release tiny membrane-enclosed spheres called matrix vesicles. These bud off directly from the osteoblast cell membrane.
And these vesicles are loaded with a specific enzyme.
Yes, they're packed with alkaline phosphatase. Inside the protected environment of the vesicle, alkaline phosphatase activity dramatically increases the local concentration of phosphate ions.
TXPO43.
Ah, so you're concentrating both the key ingredients, calcium via osteocalcin and phosphate via the vesicles. Precisely. By concentrating both calcium and phosphate within this protected microenvironment, the matrix vesicles become the perfect nucleation sites. They are the initial foci where the very first tiny crystals of hydroxyapatite begin to form.
Like seed crystals.
Exactly like seed crystals.
Yeah.
Once these initial crystals form, they grow rapidly by accretion, adding more ions from the surrounding fluid, and eventually they merge together. As they grow, they embed the type I collagen fibers and completely calcify the osteoid, transforming it into hard bone matrix. It's a beautifully orchestrated molecular process.
That coordination, just to trigger that localized calcification, it's really quite stunning. Okay, so once an osteoblast has done its job, laid down the matrix, and initiated mineralization, what happens to it? It doesn't just stick around forever.
No, it has basically three potential fates. The most common outcome is apoptosis-programmed cell death. They've done their job, and they're removed. A second group, often overlooked, sort of flattens out. They become quiescent, thin cells called bone lining cells. These cover the bone surfaces where active remodeling isn't currently happening. They're like dormant guardians.
And the third group?
The third, and perhaps most vital group for the long term, are the osteoblasts that get completely surrounded and trapped by the matrix they just secreted and mineralized. These cells differentiate into the next cell type we need to discuss, the osteocytes. They're destined to live out their lives encased within tiny chambers called lacunae.
Before we move to osteocytes, let's touch on the clinical side regarding those progenitor cells. While primary bone cancer, osteosarcoma, arising from these osteoprogenitor cells is relatively rare,
It is, thankfully, but
the skeleton is a disturbingly common site for secondary cancers, for metastases.
yes, that's a crucial point. Cancers that originate elsewhere, commonly in the lung, breast, prostate, thyroid, kidney, often migrate to bone tissue. They travel via the bloodstream or lymphatic system.
Why bone specifically? Is it just random seeding?
It seems not entirely random. The bone environment is actually quite conducive to tumor growth. It's highly vascularized, meaning good access for traveling cells. Plus, the constant dynamic turnover, the resorption and formation cycles driven by osteoclasts and osteoblasts, releases a rich soup of growth factors into the local environment. These factors can unfortunately provide a fertile ground, encouraging metastatic cells to settle, survive, and proliferate. Hashtag osteocytes, the maintainers and sensors.
Okay, so some osteoblasts become osteocytes, the mature bone cells, entombed within their lacunae. Given that they're trapped in this hard, calcified matrix with no direct blood supply, their survival hinges entirely on that communication network you mentioned earlier.
It really is a marvel of microengineering. Each osteocyte isn't just sitting passively in its lacuna. It extends numerous long, incredibly slender dendritic processes. We're talking maybe 250 to 300 nanometers in diameter tiny tentacles.
And where do these processes go?
They thread their way through narrow tunnels called canaliculi. These canaliculi radiate outwards from each lacuna, like microscopic channels burrowing through the solid bone matrix. And the critical part is the tips of these dendritic processes make contact with the processes of adjacent osteocytes and also with cells on the bone surface, like osteoblasts and bone lining cells.
How do they connect?
Through gap junctions. These junctions allow direct cell-to-cell communication and, crucially, the passage of small molecules, nutrients, ions, signaling molecules, waste products. This vast, interconnected network, the lacunar canalicular system, is their lifeline.
So it's like a microscopic plumbing and communication grid.
Exactly. It's the conduit that allows nutrients and metabolites, which originally diffused from the distant blood vessels in the central canals of osteons or from the surface, to reach every single cell buried deep within the mineralized matrix. And it allows waste to travel out.
So they're the maintenance crew, keeping their local patch of matrix healthy. But you mentioned their most crucial role, the one that really governs the skeleton's overall shape and density, is their function as mechanosensors.
Yes, this is absolutely fundamental. This is the cellular basis for the mechanostat concept, the idea that bone adapts its structure to the mechanical loads it experiences. Osteocytes are exquisitely sensitive to the movement, the flow of the interstitial fluid within those tiny canaliculi. Every time you put weight on a bone, walk, run, lift something, you create pressure gradients that push this fluid around.
And the osteocytes feel that flow.
They feel the sheer stress generated by that fluid flow against their cell membranes and processes. They can detect changes in strain, frequency, and magnitude of loading. They can even sense microdamage within the matrix.
Okay, so they sense the load. What do they do with that information?
They translate that mechanical signal into biochemical signals. If they sense increased loading or strain consistently, they send signals. We're still working out all the details, but molecules like sclerostin are involved that ultimately stimulate osteoblast activity. Basically, they tell the builders, we need reinforcement here, lay down more bone.
And if they sense decreased load, like in prolonged bed rest or spaceflight?
Then they signal the opposite. They send signals that inhibit osteoblasts and, importantly, promote the formation and activity of osteoclasts. The message is, this bone isn't being used much, it's metabolically expensive to maintain, let's trim it down.
So it's this osteocyte network that really dictates Wolff's law that bone adapts to load. Why isn't the response immediate, though? Why does it take time for density to change?
That's a great question. There seems to be a built-in regulatory delay, probably for stability. It's not just about a single instance of load, but the pattern, the frequency, and the duration of the loading changes. It takes sustained changes over days or weeks for the osteocytes' collective signaling to really ramp up or down the activity of the osteoblasts and osteoclasts significantly enough to cause measurable changes in bone mass.
And their death is a signal, too.
Absolutely. If osteocytes die, maybe due to significant damage or lack of nutrients, that absence itself is a potent signal for localized bone resorption. It confirms they must be actively maintaining the matrix around them constantly. Hashtag tag osteoclasts removers.
All right, so we have the builders, osteoblasts, and the maintainer sensors, osteocytes. Now, for the demolition experts, the osteoclasts, these are the large modal multinucleated cells responsible for breaking down or resorbing the bone matrix.
And their origin story is completely different. They don't come from the mesenchymal lineage like osteoblasts. Instead, they are formed by the fusion of multiple precursor cells derived from the bone marrow specifically, from the monocytomacrophage lineage. That's why they're so large and have multiple nuclei.
And because they're the ultimate destroyers of bone, their activity must be under extremely tight control.
Incredibly tight. Uncontrolled resorption would be catastrophic. And interestingly, this regulation often involves the osteoblasts acting as intermediaries. Osteoclast formation and activation require specific signaling molecules. Two key ones are MCSF, macrophage colony stimulating factor, and, crucially, ANGL.
ANGL.
Receptor activator of nuclear factor kappa B ligand. It's a mouthful, but it's vital. And both MCSF and ANGL are produced by osteoblasts and other nearby stromal cells.
So the osteoblast gets a signal, maybe from a hormone, and then it tells the osteoclast precursors to form and activate.
Precisely. For example, parathyroid hormone, PTH, which we'll discuss later, binds to receptors on osteoblasts. This binding stimulates the osteoblasts to increase their production and surface expression of rank. Rank then binds to its receptor, rank, on osteoclast precursors, driving their differentiation and fusion into mature, active osteoclasts. The osteoblast essentially gives the osteoclast the green light.
Okay, so they're formed and activated. How do they actually dissolve solid, mineralized bone?
The mechanism is quite remarkable, isn't it? They create a sort of sealed-off chemical attack zone.
It's absolutely incredible. It's like a subcellular acid etching factory. When an osteoclast becomes active on a bone surface, first it adheres very tightly. It forms a distinct circular ring of adhesion around the target area called the sealing zone. This involves specific proteins called integrins binding to proteins in the bone matrix.
Creating a contained microenvironment underneath. Exactly, a completely isolated resorption compartment. Within this sealed zone, the osteoclast's plasma membrane dramatically increases its surface area by folding into a complex, deeply convoluted structure known as the ruffled border. Think of it like deep folds or frills.
Increasing the surface area for action.
Precisely. And into this highly specialized, sealed pocket beneath the ruffled border, the osteoclast employs sophisticated proton pumps, specifically vacuolar type H plus AT pace pumps located in the ruffled border membrane. These pumps actively transport protons, H plus ions, from the osteoclast cytoplasm into the resorption compartment. This massive pumping action dramatically acidifies the localized environment, lowering the pH down to around 4.5.
That's incredibly acidic. And that acidity dissolves the mineral part.
Yes, the low pH is perfect for dissolving the inorganic mineral phase, the hydroxyapatite crystals.
Okay, mineral gone. What about the organic matrix, the collagen?
Simultaneously, the osteoclast releases powerful lysosomal enzymes into the same compartment. These include acid hydrolases and, crucially, matrix metalloproteinases, MMPs, like collagenase. These enzymes efficiently digest the now exposed organic matrix components, primarily the type I collagen fibers.
So it's a two-pronged attack. Acid dissolves the mineral, enzymes digest the protein.
That's it. The breakdown products, calcium, phosphate, amino acids, are then endocytosed, taken up by the osteoclast, transported across the cell, and released into the nearby capillaries. This whole process etches out characteristic depressions on the bone surface where the osteoclast was active, known as resorption lacunae or sometimes Hauschup's lacunae.
Wow. That system is so specialized, it seems like if any part of it goes wrong, the consequences could be severe.
They certainly can be. A classic example of this is a group of genetic disorders collectively called osteopetrosis, sometimes descriptively known as marble bone disease.
What happens there?
In many forms of osteopetrosis, the osteoclasts themselves are defective. Often, the defect lies in the proton pumps or the necessary chloride channels that work alongside them to maintain electrical neutrality during proton pumping. If the osteoclasts can't properly acidify the resorption compartment, they simply can't break down bone effectively. Resorption fails or is dramatically reduced.
And the result is too much bone.
Exactly. Bone formation continues relatively normally, but old bone isn't removed. The skeleton becomes progressively denser, thicker, overgrown, and paradoxically more brittle despite the increased mass.
And that causes other problems, right?
Right.
It's not just heavy bones.
No, the consequences are devastating. The thickening bone obliterates the marrow cavities, the spaces inside bones where blood cell production, hematopoiesis, occurs. This leads to severe anemia, low white blood cell counts, increasing infection risk, and low platelet counts. Also, the overgrown bone, particularly in the skull, can compress cranial nerves passing through foramina, leading to blindness, deafness, and facial paralysis. It perfectly highlights how absolutely essential that demolition crew the osteoclast is for normal skeletal health, blood formation, and overall bodily function. The balance is critical.
Okay, we've got the matrix, we've got the cells builders, maintainers, removers. Now let's look at the larger structure they inhabit, starting with the boundary layers, the linings that wrap the outside and line the inside of bones.
Right, we have two key membranes. The external lining is the periosteum, and the internal lining is the endosteum.
Let's start outside with the periosteum. It sounds pretty tough.
It is. The periosteum is a remarkably robust two-layered membrane covering the outer surface of bones, except at articular surfaces where cartilage is.
2 layers, what are they?
The outer layer is the fibrous layer. This is composed mainly of dense connective tissue, packed with type I collagen bundles and fibroblasts. It also contains the larger blood vessels that will eventually supply the bone. This layer provides significant protection and structural integrity.
And the inner layer.
The inner layer, sitting right against the bone surface, is the cellular layer, often called the osteogenic layer. As the name suggests, this layer is rich in cells with bone-forming potential. Osteoprogenitor cells, active osteoblasts when bone is growing or repairing, and quiescent bone lining cells. This inner layer is the powerhouse for bone growth and width, repair after fracture, and general remodeling potential.
How is the periosteum actually attached to the bone? Does it just sit loosely on top?
Oh no, it's very firmly anchored. This anchoring is achieved by bundles of periosteal collagen fibers called perforating fibers, or more historically, Sharpie's fibers. These are thick collagen bundles that extend from the periosteum and penetrate deep into the outer layers of the bone matrix itself, physically stitching the periosteum down. It's a very strong connection.
And the blood vessels get in through here too.
Yes, the blood vessels traveling within the fibrous layer of the periosteum branch and penetrate the bone cortex through channels called Volkmann's canals, delivering the essential vascular supply to the osteons within the compact bone.
Okay, so the periosteum is the tough outer shell, protection, and the repair kit. What about the lining on the inside surfaces?
That's the endosteum. Compared to the periosteum, it's much thinner and more delicate. It's typically just a single layer of cells.
What kind of cells?
Similar to the inner layer of the periosteum, it contains osteoprogenitor cells, active osteoblasts during remodeling or growth, and inactive bone lining cells. There's a very sparse amount of associated connective tissue matrix.
And where exactly do we find the endosteum?
It lines all the internal surfaces within the bone. This includes the main medullary marrow cavity, the surfaces of the bony trabeculae and cancellous bone, and the linings of the Haversian central canals and Volkmann's canals within compact bone.
So it's intimately involved in internal bone processes.
Absolutely essential. It plays a critical role in internal remodeling, allowing osteoclasts access for resorption and providing osteoblasts for new bone formation on those internal surfaces. It's key for processes like the enlargement of the marrow cavity during growth and the continuous turnover of trabecular bone throughout life.
This constant push and pull between formation, osteoblasts, and resorption, osteoclasts, at these linings, periosteum and endosteum, is really where the balance can go wrong in common adult bone diseases, right? Like osteoporosis.
Osteoporosis is the textbook example of that balance tipping too far towards resorption. For various reasons, hormonal changes like postmenopausal estrogen loss being a major one, but also immobilization, certain medications, nutritional deficiencies, osteoclast activity starts to consistently outpace osteoblast activity.
So you get a net loss of bone tissue.
Exactly. There's a net loss of calcium from the skeleton and a measurable reduction in bone mineral density, or BMD. This leads to thinning of the compact bone cortex and, very significantly, loss and thinning of the trabeculae within the cancellous bone. The internal architecture weakens dramatically.
Making the bone much more susceptible to fractures.
Precisely. Fracture risk, especially in the hip, spine, and wrist, increases significantly. It becomes fragile.
And this standard clinical tool for tracking this density loss is the DEXA scan.
Yes, dual energy X-ray absorptiometry, or DEXA. It's a low-dose X-ray technique that provides a very precise measurement of the current bone mineral density, typically at the hip and spine. It's vital for diagnosing osteoporosis, assessing fracture risk, and monitoring the effectiveness of treatments aimed at restoring that crucial balance.
Okay, so stepping back from the linings and looking at the whole bone macroscopically, we can see two major types of bone tissue organization based primarily on density.
Right. First, there's the dense, solid outer part. That is compact bone, also called cortical bone. As the names imply, it's incredibly dense and solid-looking to the naked eye. It constitutes about 80% of the total bone mass in the adult skeleton. You find it forming the thick, strong walls of the diaphysis, the shaft of long bones. It also forms the thin outer protective layer covering the epiphyses, the ends of long bones, and the surfaces of flat and irregular bones. Its density provides maximum mechanical strength and protection.
And then inside that dense shell, particularly the ends of long bones within flat bones, you find a lighter, more open structure.
Exactly. That's cancellous bone, which also goes by the names trabecular bone or spongy bone. It makes up the remaining 20% or so of the total bone mass. Microscopically, it's characterized by an open internal network, a latticework of interconnecting thin, bony spicules or plates called trabeculae. These trabeculae surround spaces that are filled with bone marrow.
Where is it most prominent?
It forms the bulk of the interior of the epiphyses in long bones, and also the core of short and irregular bones. In flat bones, like those of the skull cap, it forms the inner layer sandwiched between two layers of compact bone. This specific arrangement in the skull is called the diplo.
Even though it's spongy, it's still providing strength, right?
Oh, absolutely. It might look porous, but the trabeculae aren't randomly arranged. They tend to align themselves along the lines of principal mechanical stress that the bone typically experiences. This strategic arrangement provides substantial strength and support, especially against compressive forces, but does so with much less weight than if the entire bone were solid, compact bone. It's an engineering solution for strength without excessive mass.
Okay, so we have compact and cancellous bone overall. But the real strength, especially of compact bone, comes down to its microscopic organization. And here we need to distinguish between the quickly made, temporary stuff and the strong, permanent version.
Right. The first type, developmentally, is woven bone. This is also called immature or primary bone. It's characterized by its rapid formation rate. You see it during embryonic development, during the initial stages of fracture repair, forming the hard callus, and in some pathological conditions.
What defines it histologically?
The key feature is the random, irregular arrangement of its type I collagen fibers. They're just sort of laid down haphazardly, hence the term woven. It also tends to have a higher proportion of osteocytes relative to the amount of matrix, and its mineral content is generally lower than mature bone.
Which makes it mechanically weaker.
Exactly. It forms quickly, which is useful in certain situations, but it's not as strong or resilient as mature bone. The body almost always replaces woven bone with a much more organized and stronger structure.
And that stronger structure is lamellar bone.
Yes, lamellar bone, or mature secondary bone, is the highly organized bone found throughout the adult skeleton. Its defining characteristic is its organization into multiple parallel or concentric layers called the lamellae. Each lamella is typically only about 3 to 7 micrometers thick.
And the genius here is how the collagen fibers are arranged within these layers, isn't it?
It truly is structural brilliance. Within any single lamella, the type I collagen fibers are all oriented parallel to each other, running in the same direction. But, and this is the crucial part, in each successive adjacent lamella, the orientation of the collagen fibers shifts, typically by about 90 degrees orthogonally.
That's the biological equivalent of plywood, creating cross-grained strength.
It's a perfect analogy. This alternating, cross-hatched arrangement of collagen fibers provides maximum resistance to forces coming from different directions, especially torsional or twisting forces, and shear forces. It dramatically increases the bone's overall strength and resistance to fracture compared to woven bone.
And this highly ordered structure has a visible property too.
Yes, because of this highly ordered alternating arrangement of the collagen fibers, lamellar bone exhibits a property called birefringence when viewed under polarizing light microscopy. You see alternating bright and dark bands as the polarized light interacts differently with the fibers oriented in different directions. It's a visual confirmation of that underlying structure.
Okay, so lamellar bone is the strong stuff. Now, within compact lamellar bone, there's a fundamental repeating structural unit, the osteon.
The osteon, also known as the Haversian system, is the primary structural and functional unit of mature compact bone. Think of it as a complex, roughly cylindrical structure, typically running parallel to the long axis of the bone.
What makes up an osteon?
An osteon consists of anywhere from 5 to maybe 20 concentric lamellae layers of bone matrix arranged like rings in a tree trunk. These concentric lamellae surround a central canal.
The Haversian canal.
The Haversian canal, or central canal, yes. This canal contains the osteon's essential lifelines: blood vessels, capillaries and postcapillary venules, nerve fibers, and a lining of endosteum containing osteoprogenitor cells and bone lining cells.
And the osteocytes within the osteon, how do they fit in?
The osteocytes reside in their lacunae, which are typically situated at the interfaces between adjacent concentric lamellae. Their canaliculi radiate outwards and inwards, connecting through gap junctions not only to each other, but critically, extending inwards to connect with the cells lining the central canal. This ensures that every osteocyte within the osteon has access to the nutrients diffusing from the central blood supply.
What defines the outer boundary of an osteon?
The outermost boundary of an osteon is usually marked by a distinct line called the cement line. This is a layer that's relatively poor in collagen fibers, but rich in mineral and non-collagenous proteins. It represents the interface where the resorption process stopped when the tunnel for this new osteon was being created before the new lamellae started being laid down.
How does the blood supply get between adjacent osteons? If each has its own central canal, how do they link up laterally?
That's achieved through perforating canals, also known as Volkmann's canals. These are channels that run transversely or obliquely, essentially sideways, through the compact bone. They connect the central canals of adjacent osteons to each other. They also connect the central canals to the blood vessels located in the periosteum on the outer surface and in the endosteum lining the marrow cavity on the inner surface. This creates a highly interconnected, robust, and somewhat redundant vascular network throughout the compact bone.
So when we look at a cross-section of compact bone, is all the lamellar bone organized into these perfect cylindrical osteons?
No, not entirely. Because bone is constantly being remodeled, the picture is a bit more complex. You see several types of lamellae. Firstly, yes, you see intact, well-formed osteons. But filling the spaces between these complete osteons, you find interstitial lamellae. These are irregularly shaped fragments of lamellar bone. They are essentially the remnants of older osteons that were partially destroyed and resorbed during the process of forming newer osteons. They represent the history of remodeling in that area.
Okay. Remnants of old systems. What else?
Then you have the circumferential lamellae. These are broad sheets of lamellar bone that run parallel to the outer and inner surfaces of the bone, essentially following the overall circumference. There are external circumferential lamellae located just deep to the periosteum, and internal circumferential lamellae located just superficial to the endosteum lining the marrow cavity. They provide large-scale structural reinforcement to the bone shaft as a whole.
All right, let's shift focus to how bone is actually formed in the first place, osteogenesis. This is a truly stunning process, building the entire skeleton. There are two main ways it happens, but you mentioned a fundamental rule applies to both.
Yes. The basic principle is that the body almost always lays down the quicker, weaker woven bone first. Then, relatively rapidly, this initial woven bone scaffold is remodeled and replaced by the much stronger, more organized lamellar bone. Woven bone is like the initial rough draft. Lamellar bone is the final polished version.
Okay, what's the first method of bone formation?
The first method is intramembranous ossification. This is arguably the more direct route. It involves osteoblasts differentiating directly from condensations of embryonic mesenchymal tissue. Mesenchyme is that primitive connective tissue. Because it happens within these membranes of mesenchyme, it's called intramembranous.
Which bones are formed this way?
This process forms most of the flat bones of the skeleton. Think of the bones of the skull cap, the mandible, lower jaw, the scapula, shoulder blade, and the clavicle collarbone.
How does it proceed?
It begins at specific locations within the mesenchymal membrane called ossification centers. Here, mesenchymal cells cluster, enlarge, and differentiate into osteoprogenitor cells and then into active osteoblasts. These newly formed osteoblasts start secreting osteoid, which then quickly calcifies, forming irregular spicules or trabeculae of woven bone. These spicules grow larger and fuse together, trapping blood vessels and forming an initial network of cancellous bone. As this process continues outwards from the ossification center, the surrounding mesenchyma condenses to form the periosteum on the outside and the endosteum on the internal surfaces. Eventually, the outer and inner layers are remodeled into compact bone, sandwiching the cancellous bone in the middle, as seen in skull bones.
And we can actually see evidence of this incomplete process in newborns, can't we?
Yeah. Absolutely. The fontanelles, the soft spots on an infant's skull, are simply regions of fibrous connective tissue derived from the original mesenchyme, where the intramembranous ossification process, spreading from different centers, has not yet fully met and fused. They allow for flexibility during birth and accommodate rapid brain growth. They eventually close as ossification completes.
Now, this process of building bone, particularly the collagen component laid down by osteoblasts, is directly implicated in a serious genetic disorder, osteogenesis imperfecta.
Yes, osteogenesis imperfecta, often called brittle bone disease, is a tragic illustration of how critical type I collagen is for bone's structural integrity. It's a group of inherited disorders caused by genetic defects that lead to osteoblasts producing either insufficient amounts of type I collagen or structurally abnormal type I collagen.
So the organic matrix is flawed from the start.
Exactly. Without that proper collagen framework providing the necessary resiliency and pliability, the bones are extremely fragile. They lack the ability to resist normal bending forces and stresses, leading to recurrent fractures, often from minimal trauma, along with other connective tissue problems. It underscores the importance of that organic component.
Okay, so that's intramembranous ossification. What's the second, more complex method, the one used for most long bones?
That is endochondral ossification. As the name suggests, endo meaning within, chondral meaning cartilage, this process uses a pre-existing model or scaffold made of hyaline cartilage. It's an indirect method where cartilage is first formed and then subsequently replaced by bone. This is how the vast majority of bones in the body are formed, including all the long bones of the limbs, the vertebrae, and the base of the skull.
How does it kick off?
It begins with a miniature hyaline cartilage model of the future bone, formed during embryonic development. The first key event happens around the middle of this cartilage model, in the region that will become the diaphysis shaft. The surrounding perichondrium, cartilage membrane, starts to differentiate into a periosteum. Osteoblasts arise within this new periosteum and lay down a thin layer of bone around the cartilage shaft. This is called the periosteal bone collar.
And this collar has immediate consequences for the cartilage inside.
Yes, critically. The bone collar restricts the diffusion of nutrients to the underlying cartilage chondrocytes. Starved of oxygen and nutrients, these central chondrocytes swell up, they hypertrophy, and they begin to alter the matrix around them. They start producing alkaline phosphatase and matrix vesicles, leading to the calcification of the surrounding cartilage matrix.
And then these hypertrophied chondrocytes die.
Yes, they undergo apoptosis, programmed cell death, leaving behind an enlarged empty lacuna surrounded by calcified cartilage matrix.
And this becomes the focal point for bone invasion.
Precisely. This region of dying chondrocytes and calcified matrix in the center of the diaphysis becomes the primary ossification center. Blood vessels from the periosteum carrying osteoprogenitor cells and hematopoietic stem cells invade this central cavity. The osteoprogenitor cells differentiate into osteoblasts. These osteoblasts then use the remaining spicules of calcified cartilage matrix as a scaffold upon which they start depositing osteoid, which quickly mineralizes into woven bone. Bone formation then spreads towards the ends of the diaphysis, while osteoclasts simultaneously begin carving out the central medullary cavity.
So that establishes bone in the shaft.
Whatever.
But the ends of the bone, the epiphyses.
A similar process occurs later in the epiphyses, typically around the time of birth or shortly after. Blood vessels and osteoprogenitor cells invade the cartilage centers of the epiphyses, forming secondary ossification centers. Bone formation proceeds outwards from these centers, replacing the cartilage, again using calcified cartilage remnants as a scaffold.
So eventually, almost all the cartilage is replaced by bone, but not quite all.
That's correct. Two important regions of the original hyaline cartilage model persist into childhood and adolescence. One is the articular cartilage, which covers the joint surfaces of the epiphyses throughout life, providing a smooth, low-friction surface for articulation. It never ossifies. The other critical region is the epiphyseal plate, also known as the growth plate or physis. This is a transverse disc of hyaline cartilage located between the primary ossification center in the diaphysis and the secondary ossification center in the epiphysis. This plate is absolutely essential for the longitudinal growth lengthening of long bones. It remains active until skeletal maturity is reached, typically in the late teens or early 20s, at which point it ossifies completely, epiphyseal fusion or closure, and longitudinal growth stops.
Okay, to truly achieve that elite mastery of bone development, we absolutely need to zoom in on that epiphyseal growth plate. It's the engine of longitudinal growth. You described it as a kind of cellular conveyor belt. Let's walk through the sequence of events, the distinct zones we see histologically, moving from the epiphyseal side towards the diaphyseal side.
Absolutely. It's a beautifully organized structure with distinct zones of activity. Starting nearest the epiphysis, we have Zone 1, the zone of reserve cartilage. This zone consists of fairly typical-looking hyaline cartilage with scattered chondrocytes. These cells appear relatively inactive or quiescent. Their main role seems to be anchoring the epiphyseal plate to the bony epiphysis and serving as a reservoir of stem-like progenitor cells for the zones below.
Okay, the reserve pool. Then things start happening.
Yes, moving towards the diaphysis, we enter Zone 2, the zone of proliferation. Here, the chondrocytes undergo rapid mitosis. They divide quickly. Crucially, as they divide, they begin to organize themselves into distinct parallel columns or stacks, like neat piles of coins aligned parallel to the long axis of the bone. They are also actively secreting extracellular matrix, rich in type 2 collagen and proteoglycans, which increases the length of these columns. The rate of cell division in this zone is a primary determinant of how fast the bone grows in length.
So they stack up, pushing the epiphysis away from the diaphysis. What's the next zone?
Next is Zone 3, the zone of hypertrophy. The chondrocytes in these columns stop dividing and instead begin to mature and enlarge dramatically. They swell up, sometimes reaching 5 to 10 times their original volume. This hypertrophy further compresses the matrix between the columns into thin, linear septa. These hypertrophic chondrocytes are terminally differentiating. They alter their synthetic activity, notably producing type X collagen, which is thought to help stabilize and prepare the matrix for calcification. They also prepare for their own demise.
They're essentially preparing the scaffold and then initiating their own removal.
Pretty much. That leads us to zone 4, the zone of calcified cartilage. Here, the hypertrophic chondrocytes undergo apoptosis, programmed cell death. As they die, they release factors, including matrix vesicles and possibly alkaline phosphatase, that trigger the rapid mineralization and calcification of the thin cartilage septa surrounding their now empty lacunae. So you're left with hardened spicules of calcified cartilage matrix.
And this calcified scaffold is the target for the final stage.
Exactly. Zone 5 is the zone of ossification, or resorption ossification. This is where the actual transition to bone occurs. Capillaries and osteoprogenitor cells from the adjacent diaphyseal marrow cavity invade the spaces left vacant by the dead chondrocytes. Osteoclasts may remove some of the calcified cartilage, but importantly, osteoprogenitor cells differentiate into osteoblasts. These osteoblasts settle onto the surfaces of the remaining calcified cartilage spicules, using them as a framework, and begin secreting osteoid. This osteoid then mineralizes, forming woven bone directly onto the cartilage remnants.
So you get these mixed spicules of calcified cartilage core covered by woven bone.
Precisely. And this newly formed woven bone is itself very quickly remodeled by osteoclasts and osteoblasts into stronger lamellar bone, eventually incorporating it fully into the structure of the growing diaphysis.
So the summary insight here is really powerful. Longitudinal growth happens because the rate of new cartilage production through proliferation in zone 2 is normally balanced by the rate of cartilage destruction and replacement by bone in zone 5.
Exactly. The epiphyseal plate itself maintains a relatively constant thickness during growth, but because new cartilage is added on the epiphyseal side, while old cartilage is simultaneously removed and replaced by bone on the diaphyseal side, the entire plate effectively gets pushed further and further away from the center of the diaphysis. This displacement lengthens the bone shaft.
A continuous, coordinated process. And while this is happening, the bone is also growing wider, right?
Yes. Growth in length via the epiphyseal plate is accompanied by growth in diameter, or appositional growth. This occurs primarily through the activity of osteoblasts in the cellular layer of the periosteum. They lay down new bone on the external surface, adding circumferential lamellae and increasing the bone's overall width.
Doesn't that make the bone incredibly thick and heavy?
It would, except that this external addition is usually coupled with resorption on the internal surface. Osteoclasts originating from the endosteum actively resorb bone lining the medullary cavity. This enlarges the marrow cavity at a rate proportional to the external growth, ensuring the bone increases in diameter and strength without becoming excessively massive or dense. It maintains appropriate cortical thickness. Hashtag 3.3 remodeling and repair.
So even after growth stops, the skeleton isn't finished. It's not static. In adults, there's this continuous process of bone remodeling. What's the scale of this?
It's quite significant. Estimates suggest that about 5% to 10% of the total adult skeletal mass is turned over, resorbed, and replaced each year through remodeling. This ensures the skeleton remains mechanically sound, adapts to changing stresses, allows for repair of microdamage, and importantly, plays a role in maintaining calcium homeostasis. It keeps the tissue plastic and alive.
How does this remodeling happen in compact bone, where we have those dense osteons?
In compact bone, remodeling primarily occurs through the formation of new osteons, replacing older ones. It's a highly coordinated, localized process involving both osteoclasts and osteoblasts working together in what's sometimes called a bone remodeling unit, or basic multicellular unit, BMU. It typically starts with osteoclasts. They gather together and act like a motorized drill, tunneling through the existing compact bone. This advancing group of osteoclasts is often referred to as the cutting cone. They resorb both the old osteum bone and any interstitial lamellae in their path, creating a cylindrical tunnel.
And then the builders follow.
Exactly. Following closely behind the cutting cone, the newly formed tunnel is invaded by a capillary loop and accompanying mesenchymal cells, including osteoprogenitor cells. These progenitors differentiate into osteoblasts, which then line the walls of the tunnel. This trailing region of osteoblasts actively depositing new matrix is called the closing cone. They begin cyclically secreting new concentric lamellae of bone matrix, working from the outside of the tunnel inwards. This process gradually narrows the diameter of the tunnel until only the small central haversian canal containing the blood vessel and nerves remains. The result is a brand new, complete osteon replacing the older bone tissue.
This constant tunneling and refilling explains the interstitial lamellae we see the leftovers from previous cycles.
Precisely. They are the unavoidable remnants of this continuous turnover process.
And this plasticity is what allows bone to adapt, like in orthodontics.
Orthodontics is a perfect practical example. The sustained, gentle pressure applied by braces creates areas of compression on one side of the tooth root in its socket and areas of tension on the other side. This differential pressure stimulates localized remodeling. On the compression side, osteoclast activity is increased, resorbing bone and allowing the tooth to move. On the tension side, osteoblast activity is stimulated, laying down new bone to fill the space. This coordinated resorption and formation allows the teeth to be repositioned within the jawbone over time.
Can we actually measure this remodeling activity clinically?
Yes, there are ways. One classic technique, often used diagnostically or in research, is tetracycline labeling. Tetracycline is an antibiotic, but it has a useful property. It binds avidly to calcium ions. Because of this, when tetracycline is present in the body, it gets incorporated into newly mineralizing bone matrix. Since tetracycline fluoresces yellow under UV light, it acts as a visible marker of where bone formation was actively occurring at the time the drug was administered.
So how is it used diagnostically?
A common protocol involves giving the patient 2 separate short courses of tetracycline, usually separated by a specific time interval, like 11 to 14 days. A bone biopsy is then taken, often from the iliac crest, prepared without decalcification, and viewed under a fluorescence microscope. You'll see 2 distinct fluorescent lines corresponding to the 2 times tetracycline was given. By measuring the distance between these 2 lines, clinicians can calculate the exact rate of new bone apposition and mineralization rate per day. This is invaluable for diagnosing conditions where mineralization is impaired, such as osteomalacia, the adult equivalent of rickets, where you might see wide osteoid seams, but the distance between the labels is reduced, indicating slow mineralization.
Fascinating. Okay, finally in this section, let's look at what happens when bone breaks fracture repair. It seems like it recapitulates some of the developmental processes, but on an accelerated timeline.
It really does. Fracture repair is a complex but generally predictable multi-stage process that aims to restore the bone's structural integrity. It essentially compresses aspects of both intramembranous and endochondral ossification, along with remodeling, into a period of weeks to months.
What are the main stages?
Traditionally, we think of 4 overlapping stages. Stage 1 is hematoma formation. The fracture itself inevitably ruptures blood vessels within the bone, the periosteum, and surrounding tissues. This causes bleeding, and a large blood clot, the fracture hematoma, forms at the fracture site. This clot fills the gap, stabilizes the ends somewhat, and brings in inflammatory cells. Macrophages arrive quickly to start cleaning up debris and dead tissue.
Okay, the immediate aftermath. What's next?
Stage 2 is fibrocartilaginous callus formation, often called the soft callus stage. Within a few days, the hematoma is gradually replaced by granulation tissue. Importantly, cells from the periosteum and endosteum near the fracture proliferate rapidly. Fibroblasts produce collagen fibers, and some progenitor cells differentiate into chondroblasts, producing fibrocartilage. This creates a soft, pliable mass of fibrocartilaginous tissue that loosely bridges the fracture gap, providing some initial stability.
So it's like a temporary flexible splint, then it hardens.
Yes. Stage 3 is bony callus formation, or the hard callus stage. Over the next few weeks, the soft callus is gradually replaced by immature woven bone. This happens through processes resembling both intramembranous ossification, osteoblasts arising directly from the periosteum lay-down bone on the outer edges, and endochondral ossification. The fibrocartilage within the callus hypophyse calcifies and is replaced by bone, similar to the epiphyseal plate. This results in a large, irregular mass of woven bone, the hard callus that firmly unites the broken ends. It's strong, but bulky and disorganized.
So the bone is united, but not back to its original shape or strength yet.
Exactly. That requires the final and often longest phase. Stage 4, bone remodeling. Over the subsequent months, or even years, the hard callus is subjected to the mechanical stresses of returning activity and weight bearing. This triggers extensive remodeling. Osteoclasts resorb the excess bulky bone of the callus, while osteoblasts lay down new, stronger lamellar bone, aligning it according to the lines of stress, following Wolff's law. The medullary cavity is reestablished. Gradually, the bone is reshaped, restoring its original contours and strength, sometimes so effectively that evidence of the fracture nearly disappears on X-ray.
We've spent a lot of time on the structure and dynamic, but we started by mentioning the skeleton's crucial metabolic role, particularly as a calcium reservoir. Let's revisit that. Just how important is calcium?
Oh, it's absolutely vital. Calcium ions, text 2 plus year, are involved in an incredible array of fundamental cellular processes. We're talking about muscle contraction, nerve impulse transmission, enzyme activity, cell adhesion, cytoskeletal movements, membrane permeability, blood clotting, hormone secretion via exocytosis. The list goes on and on. Maintaining a very stable, very narrow concentration of ionized calcium in the blood and extracellular fluid is therefore critical for life. And the skeleton is the body's bank account for this. It stores about 99% of the body's total calcium.
So the bone acts as a buffer, releasing or storing calcium to keep blood levels just right.
Precisely. That normal range for blood calcium is tightly regulated, typically around 9 to 10 milligrams per deciliter. Even small deviations can cause serious problems. This regulation involves a constant dynamic interchange of calcium between the bone matrix and the blood.
Which parts of the bone are most involved in this rapid exchange?
While all bone contributes, the cancellous trabecular bone plays a particularly important role in rapid calcium exchange because of its incredibly high surface area exposed to the bone marrow and blood supply. The ions on the surface of those trabeculae, especially near that hydrated layer we talked about, are more readily available for quick release or uptake compared to calcium buried deep within compact bone osteons.
And this exchange isn't just passive diffusion, it's under strict hormonal control. There are two major players working in opposition, right?
Yes. Two key hormones orchestrate this balance: parathyroid hormone, PTH, and calcitonin.
Let's start with PTH. Where does it come from and what does it do?
Parathyroid hormone, PTH, is secreted by the small parathyroid glands located usually on the posterior surface of the thyroid gland in the neck. Its release is triggered primarily by a decrease in blood calcium levels. PTH is the main hormone responsible for raising low blood calcium back to the normal range.
How does it act on bone to achieve this? You mentioned earlier its action is indirect.
That's the fascinating part. Osteoclasts, the cells that actually resorb bone to release calcium, don't really have significant PTH receptors. Instead, PTH binds to receptors located primarily on osteoblasts and their precursors.
So PTH tells the builders to activate the destroyers.
In essence, yes. When PTH binds to osteoblasts, it stimulates them to produce and express RNKL, that key osteoclast activating factor we discussed, and inhibits their production of osteoprotogerin, OPG, which is a decoy receptor that normally blocks RNKL. The net effect is an increase in RNKL signaling to osteoclast precursors, leading to increased formation, differentiation, and activity of osteoclasts. These activated osteoclasts then resorb bone matrix, releasing calcium and phosphate into the bloodstream, thus raising blood calcium levels.
PTH also acts on the kidneys to increase calcium reabsorption and stimulate vitamin D activation, further boosting calcium levels.
Okay, so PTH raises calcium. What about the opposing hormone, calcitonin? Calcitonin is produced by the parafollicular cells, or C cells, within the thyroid gland. Its release is stimulated primarily by an increase in blood calcium levels. Its main action is to reduce elevated blood calcium.
How does it work? Does it also talk to osteoblasts?
No, calcitonin acts directly on osteoclasts. Osteoclasts have calcitonin receptors. When calcitonin binds, it rapidly inhibits osteoclast motility and activity, effectively putting the brakes on bone resorption. This slows down the release of calcium from bone into the blood, helping to lower high blood calcium levels. It might also promote calcium excretion by the kidneys.
So PTH and calcitonin provide this fine-tuned feedback loop to keep blood calcium stable.
Exactly. Though in humans, the physiological role of calcitonin in moment-to-moment calcium regulation seems less critical than that of PTH. PTH is definitely the dominant player in preventing low calcium, hypocalcemia.
We also need to quickly mention hormones related to overall body growth, which obviously heavily involves the skeleton, especially during youth.
Absolutely. The primary driver of postnatal growth is growth hormone, GH, also known as somatotropin, which is released from the anterior pituitary gland.
Does GH act directly on the growth plates?
Mostly indirectly. While bone cells might have some GH receptors, the major growth-promoting effects of GH are mediated through another factor. GH stimulates the liver and other tissues to produce insulin-like growth factor 1, IGF-1, which was historically called somatomedin C. It's primarily IGF-1 that acts directly on the chondrocytes in the epiphyseal growth plates, stimulating their proliferation in the zone of proliferation and maturation, thereby driving longitudinal bone growth.
Yeah. And imbalances in GH during development have dramatic effects.
Very dramatic. A deficiency of GH in childhood leads to pituitary dwarfism, characterized by short stature but relatively normal body proportions. Conversely, an excess of GH before the epiphyseal plates close, before skeletal maturity, causes gigantism, resulting in excessively tall stature.
What if there's excess GH after the plates have closed?
If GH excess occurs in adulthood, after the epiphyseal plates have fused, the long bones can no longer grow in length. However, bones can still grow in width through appositional growth from the periosteum. This condition is called acromegaly. It leads to characteristic features like enlargement of the hands, feet, jaw, prognathism, forehead, and thickening of soft tissues. It highlights that even adult bone retains some capacity for growth response, albeit differently than growing bone.
Okay, we've covered bone tissue itself in incredible detail, but the skeleton's function relies entirely on how these bones connect and interact at joints or articulations. How do we generally classify joints?
Joints are typically classified based on the type of tissue connecting the bones and functionally by the degree of movement they allow. Broadly, we can talk about synarthroses, immovable or slightly movable, and diarthroses, freely movable.
Let's start with the synarthroses, the ones with limited or no movement. What holds them together?
The synarthroses can be linked by different tissues. If bones are directly fused by bone tissue, allowing essentially no movement, it's called a synostosis. A classic example is the fusion of cranial sutures. In older adults, they start as fibrous joints, sutures, and eventually ossify.
Okay, fused by bone. What if they're joined by connective tissue?
If bones are joined solely by dense connective tissue like ligaments or an interosseous membrane, it's called a syndesmosis. These allow for a very small amount of movement. A good example is the joint between the distal ends of the tibia and fibula.
And the third type uses cartilage.
Yes. If bones are joined by cartilage, it's a synchondrosis, using hyaline cartilage, like the epiphyseal plate, which is temporary, or a symphysis. Symphyses are characterized by having a pad or disc of tough fiber cartilage positioned between the articular surfaces of the bones. These allow for slight compression and movement.
Where do we find symphyses?
They are typically found in the midline of the body. Key examples include the pubic symphysis connecting the two halves of the pelvis anteriorly, and crucially, the intervertebral discs located between the bodies of adjacent vertebrae.
Ah, the intervertebral discs. These are really important symphyses, structurally complex and vital for spinal function. Let's unpack their structure.
They are indeed phenomenal structures, perfectly designed for shock absorption, flexibility, and resisting complex forces on the spine. Each disc has two main parts. The outer part is the annulus fibrosus. This is a tough, ring-like structure composed of multiple concentric layers or laminae of fibrocartilage. The key feature here, similar to lamellar bone, is that the collagen bundles, mostly type I collagen, within each successive lamina are arranged orthogonally. They run at oblique angles, roughly 90 degrees to the fibers in the adjacent lamina.
Giving it that plywood-like strength again.
Exactly. This alternating arrangement provides incredible tensile strength and makes the annulus highly resistant to distraction, shear, and especially torsional twisting forces.
And inside this tough outer ring?
Inside the annulus is the nucleus pulposus. This is the inner central core of the disc. It's a quite different material, a viscous gel-like substance that's rich in water, hyaluronan, a large proteoglycan, and type 2 collagen, more typical of cartilage. Interestingly, it contains scattered cells that are remnants of the embryonic notochord.
What's the function of this gel-like core?
The nucleus pulposus is highly hydrated and acts like a hydrostatic ball bearing. Under compressive loads, like standing or lifting, it distributes the pressure evenly across the vertebral end plates and outwards against the annulus fibrosus. It's the primary shock absorber of the spine.
So when people talk about a slipped or herniated disc, what's actually happening structurally?
A herniated disc occurs when the tough outer annulus fibrosus weakens or tears, usually due to age-related degeneration, collagen loss, dehydration, or acute trauma. This allows the pressurized gelatinous nucleus pulposus to bulge or extrude outwards through the defect in the annulus.
And it often pushes backwards.
Yes, herniations most commonly occur in the posterolateral direction because the annulus is naturally a bit thinner and less supported there, and the posterior longitudinal ligament running down the back of the vertebral bodies tends to push escaping material sideways.
Why is that a problem?
The problem is that the spinal cord and spinal nerve roots lie directly behind the intervertebral discs. When the nucleus pulposus herniates posteriorly or posterolaterally, it can directly compress these neural structures. This compression can cause significant pain, often radiating along the path of the affected nerve, like sciatica, numbness, tingling, and muscle weakness in the areas supplied by that nerve. Hashtag tag tag 4.3 diarthroses. Synovial joints free movement.
Okay, so we've covered the immovable and slightly movable joints. Now for the ones that allow the most motion, the diarthroses, commonly known as synovial joints. These are the joints like our knees, elbows, shoulders, hips.
Exactly. Diarthroses are characterized by allowing free movement between the articulating bones. They are structurally more complex than synarthroses. They typically unite long bones. Key features include the ends of the bones are covered by smooth articular cartilage. The bones are held together and aligned by ligaments, and a fibrous articular capsule that encloses the entire joint.
And inside that capsule,
The capsule defines a space called the joint cavity, or synovial cavity. This cavity is filled with a small amount of viscous lubricating fluid called synovial fluid.
let's look at the articular cartilage covering the bone ends first. What type of cartilage is it, and what's special about it?
Articular cartilage is hyaline cartilage, but it has some unique properties. Most notably, unlike most other hyaline cartilage, like in the trachea or nose, it completely lacks a perichondrium. This means it has no direct blood supply or nerve supply and limited capacity for repair if damaged.
How does it get nutrients then?
It relies entirely on the synovial fluid for nutrients and oxygen and for waste removal. These substances diffuse from the synovial fluid into the cartilage matrix. This diffusion process is aided by the intermittent compression and decompression of the cartilage during joint movement, which helps to pump fluid in and out of the matrix.
And the structure of the collagen fibers is specialized too.
Yes, the type 2 collagen fibers within the articular cartilage matrix are arranged in a very specific pattern, often described as forming arches or arcades, the Benninghoff arcades. In the deepest zone, near the bone, the fibers run perpendicular to the surface, anchoring the cartilage. In the middle zone, they curve to run more obliquely or randomly. And in the superficial zone, closest to the joint cavity, they run parallel to the articular surface. This architecture is thought to be optimal for distributing compressive forces evenly across the joint surface and resisting shear stresses.
Given its avascular nature and reliance on synovial fluid, damage seems like a big problem. This is where conditions like arthritis come in.
Precisely. Osteoarthritis involves the gradual wear and tear and degeneration of articular cartilage. But rheumatoid arthritis, RA, is different. It's a systemic autoimmune disease where the primary target is the synovium, the lining of the joint capsule.
How does attacking the synovium lead to cartilage destruction?
In RA, the synovial membrane becomes chronically inflamed and thickened, forming a pannus. The inflammatory cells within this pannus, particularly activated macrophage-like cells and fibroblasts, release large amounts of destructive enzymes, including collagenases and other proteases, like MMPs, as well as inflammatory cytokines. These enzymes directly attack and degrade the adjacent articular cartilage matrix, leading to erosion of the cartilage and eventually erosion of the underlying bone. This causes severe pain, joint instability, deformity, and loss of function.
Which brings us to that crucial lining itself, the synovial membrane or synovium. It produces the fluid that lubricates and nourishes the joint. What's it made of?
The synovial membrane is a specialized type of connective tissue that lines the inner surface of the articular capsule, everywhere except over the articular cartilage itself. Importantly, it's not a true epithelium, it lacks a basement membrane. It's typically only 1 to 4 cell layers thick and sits on top of underlying loose or fibrous connective tissue that's rich in blood vessels, particularly fenestrated capillaries, capillaries with small pores, which allow fluid to filter out easily. The membrane itself contains 2 main specialized cell types, often called synoviocytes.
What are the 2 types?
Type A synoviocytes are macrophage-like cells. They are derived from blood monocytes and reside within the synovial lining. Their primary role is phagocytic. They clean up the joint cavity by removing wear and tear debris, like small fragments of cartilage, dead cells, and fibrin. They also play a role in regulating inflammation within the joint.
And the other type?
Type B synoviocytes are fibroblast-like cells. These are the primary producers of the key components of synovial fluid. They synthesize and secrete large amounts of hyaluronin, also known as hyaluronic acid, a large glycosaminoglycan, and various proteins and proteoglycans, including lubricin.
So how does the fluid itself form?
The synovial fluid is essentially an ultrafiltrate of blood plasma that leaks out from those fenestrated capillaries in the underlying tissue, mixed with the hyaluronin and other molecules secreted by the type B synoviocytes. The hyaluronin gives the fluid its characteristic high viscosity, making it an excellent lubricant to reduce friction between the articular cartilages to almost negligible levels. The fluid also serves as the vehicle for transporting nutrients like glucose and oxygen to the chondrocytes in the avascular articular cartilage and removing their waste products.
So it's both a lubricant and a nutrient delivery system produced by this specialized membrane. The interplay in joints is just as intricate as the cellular battles within the bone itself.
Absolutely. It's a beautifully integrated system designed for movement, stability, and longevity. Hashtag tag outro.
Well, this deep dive has certainly confirmed that the skeleton is anything but a static, inert frame. We've really covered the full spectrum today, from the precise molecular triggers that kick off mineralization, involving osteocalcin and those matrix vesicles, all the way to the intricate microarchitecture of lamellar bone and the osteon, and perhaps most importantly, that continuous dynamic antagonistic cellular activity, the builders versus the removers, that keeps the entire structure plastic, responsive, and fundamentally alive.
And if we try to connect all these threads to see the bigger picture, the entire musculoskeletal system emerges as just this masterpiece of adaptive engineering. Think about the synchronicity required. You've got the mechanosensory osteocytes deep inside the bone, detecting minute changes in fluid flow from everyday activity. That triggers signals, which might then influence the swift hormonal cascades involving PTH and ARE KL, driven by the body's systemic need for calcium.
It's all interconnected.
It's all interconnected, designed for immediate coordinated response to both mechanical demands and the body's crucial chemical needs. It's constantly adjusting. So maybe a final provocative thought for you, the listener, to mull over. Consider the sheer scale of continuous cellular activity occurring within your own skeleton right now as you listen. Imagine the thousands upon thousands of microscopic cutting cones, those osteoclast teams tunneling through your compact bone, and the closing cones of osteoblasts following right behind, refilling the tunnels.
That 5–10% annual turnover.
That relentless, localized turnover, replacing 5 to 10% of your entire skeleton every single year. And realize that all of this microscopic warfare, this constant resorption and formation consuming significant energy, is ultimately driven by that fundamental metabolic imperative, the need to maintain that absolutely critical 99% calcium reserve, keeping your blood levels stable so that every other fundamental life process, from your heartbeat to your muscle contractions, even your thoughts, can continue uninterrupted.
The ultimate living material, indeed. It's a powerful way to think about it. Thank you for joining us for this deep dive into the mastery of bone and joint histology.